Alrighty, welcome everyone to a new episode of Mr. Marinelli's Bio Boost. Hopefully, you're all having a lovely holidays and getting ready for the exam. But I just want to make a quick video on cellular respiration as requested by Tirana. So I'll try and make it really quick, so it's just a nice, really bare bones sort of summary of the whole process. And then if you want to look more deeply into it, you can watch things like Douchi's videos or any of the longer videos that are available on YouTube, which are all great. And obviously you can ask me any questions as well. Feel free to ask in the comments or on Teams or via email. So what I've done is I've color-coded the whole, equation to try and line it up with each of the stages. So we've got oxygen and glucose, and that's going to make carbon dioxide, water, and all-important ATP output. Now, we can see that in glycolysis, this is our first stage, okay? This is happening in the cytosol of our cells, and this is going to be the process that's going to produce just 2 ATP, but it's really fast, okay? We can do this 100 times faster than we can do cellular respiration as a whole, okay? But what are the drawbacks? Obviously, we can't do this, non-stop because we're going to build up lots of byproducts, okay? And that's where we're going to talk about fermentation. So fermentation is essentially just where we've got this glycolysis happening, but then with this, the NADH, we want to recycle that. So we convert our pyruvate, basically, to lactic acid, and we unload that NADH, so that we can have a NAD plus again and create that process again. But obviously that's going to build up lactic acid, which is then going to be poisonous, and that's not what we want. So it's good in a pinch when we need energy, but not going to be good in the long term. But the purpose of this video is to talk about cellular respiration as a whole. So glycolysis occurs first. We need our glucose, that's sugars that we get from things we eat. We need NAD plus, which is an unloaded carrier. We've got ADP, another unloaded carrier, plus our phosphate as well. Now, what we're going to do is we're going to split this glucose up. So the splitting of that is going to provide us the energy to basically produce NADH and ATP. Now, the ATP is good because we want energy. That's why we're doing this process. The NADH is going to be really important later on when we get to the electron transport chain. So keep that in mind. Then we go on to our Krebs cycle. Now, pyruvate, we see over here it says including pyruvate oxidation. You may remember that pyruvate doesn't actually move into this process directly. First, it gets converted into what's called acetyl-CoA, and then it can move into the process. But that part isn't too important. Sometimes you see this written as pyruvate. Sometimes you see it written as, I'll write it down, acetyl-CoA. Okay, so that just means basically they're acetyl groups and they're brought in by the coenzyme A. Now, essentially, this is going to occur in the matrix of the mitochondria. So we need oxygen for this process to occur. So that's an important part. Our glycolysis is anaerobic. Our Krebs cycle and our electron transport chains are aerobic. Okay, so we've moved into the matrix of the mitochondria now. You'll notice that the ATP yield for this process is still only 2. But this is where you've got to remember, and it makes sense once you look at what the inputs and outputs are, that the purpose of the Krebs cycle is to provide our loaded carriers for the electron transport chain. So you can see that pretty much all the inputs to the Krebs cycle are essentially these unloaded carriers. So what we want to produce out of them are our loaded coenzymes. Okay, so we've got NADH, FADH2, and ATP. Okay, so obviously ATP, great, we want energy, but we need these loaded carriers. Okay, now CO2 is a byproduct of this stage, so that's from the breakdown of the acetyl-CoA. So this is where our CO2 comes into it, and we can see that's one of our outputs from cellular respiration. So if you're running a lot, you breathe out. Okay, there's our CO2 leaving our system. Okay. Now we finally go into the electron transport chain. Okay. Now think about what we've created to get to this point. We've created NADH, FADH2. Okay. So those are going to be our inputs that go into our process. Okay, we can see here, we've got NADH, FADH2, and our ADPs. Now remember, we always need our ADPs plus P to create our ATP. But the other inputs make sense. We've got these unloaded carriers and we've got oxygen. So we've got these loaded carriers and we've got oxygen as well. Okay. Now that's going to become important in a second. I'm going to scroll down a little bit here. So we've got our NADH and our FADH2. Why do we need them in this stage? Well, this is where we're going to be pushing our loaded carriers through to provide energy to pump hydrogens or protons into this intermembrane space. Okay, we produce all these gradients that essentially make it really high hydrogens over here, and we get those hydrogens again from these loaded carriers becoming unloaded, and basically that provides energy because remember it's a catabolic reaction. We've got something bigger that we're making smaller. It's catabolic, it's exploding, we're creating energy. It moves and allows energy to pump hydrogen ions into this intermembrane space. Remember, if we're wanting to build up a big concentration gradient, that's obviously not going to be diffusion, that's going to be active transport. We need energy for that. So this energy is producing this big concentration gradient of hydrogen ions in this intermembrane space. Now, as we continue to build up, we see this ATP synthase. Okay? Now remember, this is almost like a propeller. So with these hydrogen ions, once they want to diffuse back into this space, because remember, diffusion will always, take precedence. It's basically going to be this process that you can't fight against. It's just passive transport. All these hydrogen ions are going to want to come back through to equalize that gradient. As they come back through, they go through this ATP synthase that basically, if you can imagine, spins the propeller, which provides energy for the ADP plus PI to be bound together. Okay? Remember, that would be an anabolic process because we are building something, okay, which means it requires energy. Okay, so all these hydrogen ions moving back through provide the energy to build all these ATP molecules. And how many do we make? We make 26 or 28. Okay, now that's really important because obviously that is our output of ATP in this section. This is why we need all of our loaded carriers, so we can break them apart to provide the energy to build the ATP. Why do we need oxygen? Why is that in this last phase? Well, we need it to bind to all these now free hydrogen ions that are coming back through to be the final acceptor. Okay, we don't want to keep having these hydrogens come through, otherwise we're not going to be able to build up the gradient. So we want to take those hydrogens away once they've been used. We do that by binding them to oxygen, and that makes water. And that is where we get this final water molecule created in cellular respiration. Okay, so let's just really go back through, I guess, the real fundamentals of this. And this is a thing I put on the board earlier. So we've got earlier in the year, so we've got glycolysis in the cytosol, where we're converting our glucose into pyruvate, our NAD plus into NADH, which is going to be important later on, and our ADP plus PI into ATP. Okay, that's the point of that process. Once we get oxygen, we can go into the Krebs cycle in the mitochondrial matrix, where we got our acetyl-CoA, which has been already converted from pyruvate. And then that's going to be our CO2. It's going to be a byproduct. Basically, that's providing us the opportunity to make NAD plus and FAD plus into our loaded electron carriers so that they can enter the electron transport chain and provide lots of energy. Once we get to the electron transport chain in the inner membrane or the cristae of the mitochondria, we basically want to split those loaded carriers into our NAD plus, our FAD plus, and that is going to produce lots of ATP. And we've got our oxygens sitting there ready to take all those extra hydrogen ions and make water. Okay, so at the end of the day, what our output is, just get out of it, is 30 or 32 ATP. So we've got oxygen plus glucose making carbon dioxide, water, and the all-important 30 or 32 ATP. Okay, it's important you say or, not a range, because it's either 30 or 32. Okay, hopefully that helps. If you have any questions, please let me know. But otherwise, have a lovely rest of the day. Catch you later.